Hi, I'm Rivera Waterman, an environmental science PhD student in the Department of Civil, Environmental, and Architectural Engineering at the University of Kansas. I'm going to present some motivation and preliminary results for a field study in a Kansas wetland that will investigate how different aquatic plant types act and interact in nutrient removal under dynamic flow conditions. Wetlands and the aquatic plants within them are recognized for their water treatment capabilities. The mechanisms and rates of nutrient removal are different across aquatic plant types, such as emergent, submerged, and floating vegetation, and are often determined with individual plant species or communities consisting of a single plant type. However, multiple plant types can co-occur within natural wetlands, and synergistic interactions between them may influence nutrient cycling, particularly under varying hydrology. Based on current literature and approaches, some relationships have been identified for the different plant types in nitrogen and phosphorus cycling specifically. Generally, emergent vegetation is more efficient at nitrogen removal, while submerged vegetation is more efficient at phosphorus removal, with the difference not as clear for floating vegetation. Thus, questions remain on the role that different plant types play in removal of these nutrients in the presence of other vegetation types, such as if there are synergies or trade-offs that occur between them, if these relationships are similar for different nutrients, and if these relationships exist, are they impacted by varying flow conditions? Briefly, we hypothesize that these relationships will generally hold for nitrogen and phosphorus removal, with emergent vegetation not as sensitive and potentially more effective at nitrogen removal under dynamic hydrologic conditions, while floating and submerged vegetation are more sensitive, especially at low flows or low water levels. To address these questions and test these hypotheses, we are conducting a field study in a wetland located northeast of Lawrence. The hydrology of the site spans from groundwater-fed stream at the inlet to a wetland buffer of emergent vegetation, then a ponded area populated with submerged and floating vegetation. The pond then outlets into a Kansas River tributary. We conducted an initial grab sampling campaign to understand spatial gradients for nitrate and soluble reactive phosphorus, or SRP, across the sites and across different vegetated regions. Results show that both nutrients vary across the site, indicating that internal biogeochemical processing is occurring as nutrients are transported from source or sources where concentrations are higher into the wetland pond region and to the outlet where concentrations are lower. These results also appear to show that there are two different source locations, with nitrate coming from the groundwater-fed inlet in the north and SRP from groundwater seepage from the northwest. We will continue to assess site water chemistry and nutrient dynamics using multi-parameter sensors located near the outlet and in the inlet, as well as continue spatial grab sampling under different conditions. With the multi-parameter sensors at the inlet and outlet, we were able to monitor internal water chemistry dynamics from a storm event in late October. Those results show that there is a negative or dilution relationship between conductivity and increased flow from rainfall. Although we are not monitoring discharge yet, we aim to use this concentration discharge relationship with high frequency sensors to monitor dynamic flow conditions, which we will couple with our internal nutrient and vegetation data. This was just preliminary work provided to gain insight and provide direction for the field study. So next steps include deployment of sensors for continuous monitoring of water chemistry and flow in the study area, continued grab sampling of water and nutrient chemistry, as well as characterizing and sampling the various vegetation types, focusing on parameters like location, abundance, density, biomass, and nutrient content of each plant type. With this data, we will develop statistical models that can inform any synergies or trade-offs in plant type nutrient removal under different conditions. The results of this research will aid in assessing ecosystem services of natural wetlands by revealing nutrient vegetation flow relationships that contribute to water quality improvements. Lastly, I want to thank Kent Dye, Hannah Min, Joey Kraft, and Joseph Hawker for helping with experiments, analysis, and sampling. And I would like to thank Steve Lehman for land access and accommodations. Thank you.